Assalamu alaikum everyone, dear participants, dear all members. We are here on the 29th CNE from this Lambda chapter as a webinar. And we are pleased to invite our mentor, Dr. Rezzan. I have no words to give her introduction. She is the legend of the dietetics. And today she is presenting on genetics and dietary factors to the variation in eating behavior. And I think this is a very different topic to listen from you, Dr. Rezzan. So the deck is to you. Over to you.
वो डेस्कटॉप में जाके शेयर करूं हाँ या डेस्कटॉप minimize what
then open it
yeah
or copy somewhere.
that's it
Yeah, that's great.
Yeah.
okay.
Over to you, Dr. Rezzan. You're most welcome.
Okay, so we can start. Thank you very much for your nice introduction. Okay, today I'll be talking about the genetics and dietary factors to the variation of eating behavior. And this topic will be very boring for some of you, and it will be very interesting for some of you. And I am sure you are going to refresh your even high school or maybe university years for the genetics. So I'll just give you briefly the genetic background, just to refresh the memories. And I'll be talking about the nutrigenomics. And this is the influence of diet on genes, how nutrients are affecting expression. And how can I complete it? And then nutrigenetics, the influence of genes on diet, how genetic makeup affects the nutrients, how they are metabolized. And I'll give a few examples for the genetic variations, influences the interaction between eating behavior, diet, and how it leads to our health conditions, and overview of the concept of metabotyping for the delivery of personalized nutrition. Genes are involved in all eating activities, including calories, meal size, and meal frequency, and our liking of food for specific macronutrients such as fat as well as proteins and carbohydrates. They all affect our genetic makeup. And you can bring something and people say, oh, that's yucky, and then the other one says it's delicious. And you shouldn't blame all the time your environment. Sometimes it's in your genes. Okay, why the interest in personalized nutrition? Because recently everybody is talking about that. There are three things for this interest. It is mainly to the practice of preventive approaches for optimizing health. And if you know our genetics, when we can do the delay in onset of the diseases, and if we already have diseases, we can work on diminishing its severity. Sounds like giving the medical nutrition therapy, right? Okay, this is the things that we can see all the time, omics and omics and omics. And then this omics are science and used in the nutrition versus health. And then how the nutrients and then nutritive components are affecting our genes and how they are affecting the health, and that can be vice versa too. So the nutrients and bioactive compounds in the food, then they affect the nutrigenomics, and then it has three steps, and then it's nutrigenetics, epigenetics, and transcriptomics, and then this will all change our expression of the genes, and then that will affect our phenotype. So in a summary, we can say that nutrigenomics are focused on the effects of the nutrients over the genome, proteome, and metabolome, how the components of a particular diet may affect the expression of genes, which may have increased its potential or which can be suppressed. So either it can cure the problem or it can make it worse. This is all in our variants. Because genetics is the study of heredity. Everybody knows that. And DNA bears our genetic code, and DNA is packed into chromosomes, and chromosomes carry our genes. And here you can see that it is just like a helix, and then it has some backbone, and then it has some nucleotides, and then it can contain the genes contain this DNA, and then it makes the difference. And then this shows how they are, this is the phosphorylated and sugar base, and sugar, you can see that it is hydroxyl ribose in the DNA and then ribose in the RNA. And when you see the middle backbone, these are the nucleotides. And then we have 4 types of nucleotides, adenine, British and Americans pronunciation is different. Either you can say adenine or adenine, and thymine, cytosine, and guanine. And when you see that A always matches with the T, and G matches with the C, and T is again combines with the A, and C always with G. And in order to remember it, then you can just say simply apple in the tree and car in the garage. See, apple in the tree, car in the garage. So this is indeed the deoxyribonucleic acid. And in the ribonucleic acid, the cytosine changes with the uracil. These are the simplified things. And DNA is packed in chromosomes, and we can see that they are all in the nucleus, and then it can go for the transcription and translation, and then it goes for the protein production. So the genome also encodes RNA and tRNA, which are involved in protein synthesis. Within the nucleus of the cell, you can see that throughout the body, there are 23 pairs of chromosomes. Within each chromosome is the genetic material organized into sequences. These are known as genes. And during the process of transcription here and the translation, proteins are formed. In transcription, the information stored in a gene's DNA is transferred to RNA in the cell nucleus, and messenger RNA carries the message from the DNA out of the nucleus into the cytoplasm. In translation, messenger RNA interacts with the ribosomes to read the sequence of the messenger RNA bases. And then it goes like that. This is useful to the cell's information in the DNA. First must be decoded and translated into the proteins. If they are not decoded, the proteins will not be formed. Okay, this is sometimes, you can see that. And we already mentioned that a chromosome is like a cookbook. A gene is like a recipe in the cookbook. And sometimes we make mistake in the recipes. And then this is called mutation. Mutation is like an error in the recipe. Even a complete lack of recipe is possible. So these are the simple things. So you can see that we have the A and B. And then here in the A, C is coupling with G. But in the next one, G is changing place with the C. So this is the variant genotype. So the protein that they are supposed to synthesize, it is changing the form. Sometimes that can be healthy protein, sometimes sometimes it spoils the health. And this affects the variations in human behavior, morphology, and even it affects the susceptibility to the diseases. And these are called SNPs, single nucleotide polymorphism. These are the small sequences, differences within the genes where the DNA sequences of many individuals vary by a single base. Okay, this is how we can read the genes. Sometimes we can read them, sequence of the pair of homologous chromosomes. And we have two types of alleles, and then these alleles are the variants in the genes, and then we have the major allele, and then we have the minor allele. And then for the major alleles, they repeat it more. You can see that A is repeated more, and then in the minor, the C is repeated less. And when we are encoding, and then when we are talking about the genes, because we'll be talking about this 0, 1, 2 types and copies and such thing. And then the major allele, we call it 0, and minor allele 1. And sometimes that can be 2 alleles too. These are the variants on the same gene. And then how the genes are named, because they have such a long name, and then it's really difficult to control them. So scientists keep track with genes by giving them unique names, because gene names can be long. Genes are also assigned symbols, which are short combinations of letters and sometimes numbers that present an abbreviated version of the gene's name, like in the previous slide I was mentioning. And different form of same gene is called allele. This is the variant in the genes, right? For example, if we have TAS2 or 38, it means this is the taste gene, and then it's on the second receptor, and then 30 member of the 38, and then it has different synapses. So these nucleotides shuffled, and then they are located in certain chromosomes, and then see they all make the amino acids. When they get together, like PAV dash to PAV, this is the major one, and then repeating the same thing, and then this is proline, alanine, valine, and that can be change the place, so that can be either with this one, this combines, or only the recessive one repeats itself, because we get one gene from each parent. Okay, so you can overcome the genetic risk by changing the lifestyle. This is a very good example for it. Specific dietary profiles can modulate the delicate balance between the health and diseases. See, this is the one. Depends on the balance. And then this is between health and disease, and acting either directly or indirectly on the gene expressions. These two things affect the gene expressions. And then we were talking about the polymorphism. And genes express themselves through proteins, enzymes, or specific proteins designed to get things started. And you can see that in this one, if you just genetic code of obesity increases the left arm of the scale. Okay, if we just do this sedentary life and then eating all those wrong things, I'm not going to give you the one by one, we are all dietitians, and then this becomes dominant, and then it can lead to obesity. How genetic mutations can lead to functional differences. This is another dilemma. So this is, for example, we can say Aisha or Fatma or Naz or Nazli, whatever you say, and they are both suffering from the mild heart attack. And they are both in the low dietary fat, and one of them is having high HDL, good cholesterol, and then the other one is having low HDL. And then this surprises all the health persons. And then the doctors, dietitians, they say, what's going on? They are having the same exercise, same diets, and everything is same. But why these things are different? Then the explanation of it is we have a hepatic lipase, and this hepatic lipase, when it has overexpression and it decreases the expression of the HDL. And then this is the polymorphism of the hepatic lipase. This is on the again 551 for transcription initiated. This is the select variation, okay? And in this place, the C is changing with T. And when we go with the CC alleles, and then because this increases the HDL, hepatic lipase, because when the hepatic lipase is increasing, it has an adverse effect. You can see that it is decreasing the good cholesterol. But when there is a polymorphism there, then CCLS on the low protein diet, they have the low HDL, but when there is polymorphism there, even they have one copy or two copy from the T, then they have an increased HDL. I hope it's clear. And then there is another gene is associated with the BMI and food intake, and then this is called the Aqua A2 gene, and then it's responds to saturated fat, and it increases the weight gain. And then this gene comes again in 3 types. That can be genotype A, B, and C. And you can see that genotype A may be more prone to excess weight gain and increased BMI with high saturated fat intake. And then the other one, copy 1 and 2, may be prone to excess weight, may not be prone to excess weight gain with high saturated fat intake. So the first one we recommend that they require the restricted caloric intake to less than certain amount, and this other group we can just tell them that okay, you just keep on this and keep it within the normal range. And this is when we are going for the impact of food selection and then cognitive behavior, you can see that dietary intake regulation may be affected by SNPs in the genes that encode taste receptors, variety of peripheral signaling peptides, they are all affecting the hormones, because the hormones, enzymes, they are all protein, and then our DNA is making this protein, and genes are making these proteins. So we already mentioned about the other two, and then I'll just go for another gene, like this is the FTO gene, and then this is related with the reduced satiety. And you inherit a copy of the gene, FTO gene from each of your parents. So you can either have 0, 1, or 2 copies of the DNA, and the difference that makes you perceive fat more intensely. If you have just 1 copy of this DNA differences, you will taste the fat flavor more strongly than if you don't have any, but not as strongly as if you had 2 copies. So if you have 2 copies of genotype FTO gene, you are at high risk to gain weight. And then if you have 1 copy, you are in medium risk. And if you don't have any copy, then you are fine. So this explains most of the people why they are gaining more weight, why they are not, although they are eating the same thing and they are doing the same thing. So this is but there is one good news that if we have the disease-creating variation of the LLE, then they did the research and they find out that when they increase the dietary intake of the protein, that may protect against the effects of the bad genes. And then this is more effective when you compare it with the non-copied genes. And then this is the abbreviation of it. Then you can say that high protein content, high protein protects the effect of risk variance in FTO. So this is the risky gene, AA, and then when they put this person on the more than 18% of the protein, and then they reduced more weight. See, percent of body weight is more than the first one. So this is the good news. So it means we can change our phenotype by changing our dietary habits and lifestyle. Actually, some of the research brings out that about 30% of the problems are genetic, and 70% we can just control it with our diet and lifestyle. Then when we can talk about the epigenetics, this epigenetics doesn't change the DNA. And this epigenetics, they affect the histone tails, acetyl group, and histones, and then this is just on the DNA. It is just like a mark that can be marked and that can be just removed. And epigenetic changes involve the placement of small modifications of DNA-based structure called marks, which alter DNA function without changing the DNA-based sequence. Epigenetic marks can also involve genetic chemical modifications of the histone proteins that package in DNA. These marks do not alter the structure of the protein. This I keep on hammering it. This is encoded by the genes. Instead, they alter the level of expression of or the circumstances under which the gene is expressed. So these are just a temporary thing, but you can just pass it from one generation to another generation too. So this is important. If the parents are having bad effects, then it affects the uterus. It affects the small children. For example, if we have two persons, one is having the harmful westernized diet, the other one is having the safe Mediterranean diet, and you can see that this westernized diet is affecting this gentleman, and then it makes it obese phenotype, and then this obese phenotype directly affects the epigenetics, and then this epigenetics affect his health. It can cause fatty liver disease, diabetes, whatever you want to say. And then it's not only affecting him, then it affects the next generation too. And then when the next generation is affected, then it causes diseases, and then again they become the obese phenotype. And then the other one, if they are going for the proper Mediterranean diet, the healthy one, and then you can see that everything is just opposite. Lean phenotype, and then delayed aging, low risk and everything, and then it affects genes, and then child, baby, and then that can be healthy. So we have to be careful. So this is epigenetics, is the study of gene activity during the development of complex organisms. So they did also research on the little small bees, and then what happens if they just keep on giving the sweet, and then whether that affects your genes, and then can it pass to another generation, or whether you can just eliminate it. So in this study, flies fed high sugary diet experience lower sweet taste sensations as a result of the decreased responses of the sweet sensory neurons to sugar stimuli, and then the reducing sweet sensation then make them take more sugar. Half of these transcriptional changes persisted despite returning the animals to a control diet and causing a permanent decrease in sweet taste. And then when the sweet taste is decreasing, then they keep on eating more. So it affects the behavior. So the sweet taste deficit develops within 2 to 3 days upon exposure to the high sugar diet and dependent on the concentration of sugar in the diet, but were independent of fat accumulation and weight gain. So the rest, again, it's up to you because of the lifestyle. Whether you can burn them or something, metabolize, again, they are related with the genes too. So food preferences are shaped during fetal development, and eating behavior develop gradually over time. So let's have something light. Okay, this child is just two to 2.5 years old and happened to be my granddaughter. And you can see that. You can see that how much she's enjoying eating lemon. This is lemon. Have you seen such a child? And then in front of her, there is salad bowl. And then she thinks that it is sour, but still she keeps on eating. See? See? Taking it more. So this is one of the examples that it develops in the small age, and it is more predominant in the children. And then this is another one. This is in my house, and then I was just giving the chocolate to her to eat, and look what she did. You can see that. How with great appetite she was taking the lemon, and then how she's throwing out the chocolate. And then, as if nothing happened, goes for the iPad. And this is the Turkish coffee. I don't know whether you are familiar. All my grandchildren taste the first coffee from my hand. And then we don't put any sugar and we don't put any milk in it. So you can see that when I give them, okay. See, this is the coffee. Look at that. First she's tasting, because the first time she's tasting this, and then open the mouth and for the second helping. So these are the living examples for the children. So how we can change our taste buds later on. So same genes have different tastes. Vegetables in the cabbage family called cruciferous, such as broccoli, Brussels sprouts, cauliflower, cabbage. These vegetables also contain several different types of natural occurring sulfur-based compounds. When you eat them, these compounds bind to a bitter taste receptors. That receptor sends a message to the brain. The brain processes that taste sensation as bitter. And depending upon the genetics, each of us responds differently to the messages of the bitterness. Those who like it maybe detecting a less intense bitter flavor and odor. Those who like it may be detecting this, and then we can just introduce it to them. Heat destroys these compounds. So maybe we can just cook it and then cook it when the lid is open of the dish cake. So we can give it to them and they can have it. And then this is the variations in the taste receptor genes which contribute to the individual differences in taste-related behaviors. And this is the genotype, combination of alleles that determine the particular traits. For example, one capital and small t is genotype for the phenylketonuria carbamide is just simply called PTC, testing traits. So this is just put it in the strip and then make the person to grade it, whether they are thinking that it is very sweet or it is very bitter or less bitter or they don't get any taste. And then it affects the phenotype. So these are the, whether they are testers, and if they have the two recessive and passive genes, LLEs, then they are the tester. And if they have two capitals, it means they are the super testers. Okay, this is what are the taste buds. We always talk about the taste buds as the dietitian, right? We always tell them, change your taste bud, change your taste bud. But we have to explain it to them in a very simple way. If a person, and these taste buds are found in the papilla, and then in this papilla, these are the bumps on the tongue, and you can see that. And if the PTC receptors are found on these taste buds, and if you have these things more often, then you can get the taste more. And then it goes for the nerve cells and transmits the signal to the brain. And if you like the taste, then your brain says that, okay, you can pursue it in a different way. And then you say, this tastes really bitter, and maybe you can just throw it and you say it's yucky, and you don't get the taste, and you can say, okay, that's delicious, and you give me more. These are the things that we have to take it into consideration. So where are the tests PTC come from? Where does testing PTC come from? So you have two copies of every gene. You get one from the mother and one from the father. And your two genes are the genotypes. And a gene can be dominant or recessive, like we were talking about the capital T and small t, and the expressed trait is the phenotype. So this determines which trait is seen, which is a person's phenotype. So testing PTC comes from our gene. You cannot see the genes, but you can just see the phenotype. So these two traits are important for determining the PTC test sensitivity. Like I'm just repeating in a simple way. PTC receptor genotype. Do you have the receptors that enable to test the PTC? And then these are the receptors. See, this has 2, this has 1, and this has none. And the density of the papilla on your tongue correlates the sensitivity of testing PTC. So super testers, they always have more papilla and then more receptors. So why can some people test PTC and others cannot? So this is the simple thing. You can see it is just like lock and key, and this is the gene and this is the receptor, and then you can just see it, and then it has to hook in this. If it doesn't hook in that, then you cannot get anything. So this is the polymorphism of the polymorphism. Synapse in the test receptor variation in genes can cause variations in receptors. Then this is the genetics, okay? And then we can see in all of them, this is too many, too few, and then these are mutated and they are the non-testers because they are not touching at all. Then when you give them something, then they keep on eating. So PTC is dominant over inability to test PTC, which is the recessive one. So this is the dominant one, TT, and then this is the recessive one, small t, and then this is TT. And then these are the genotypes, and then these are the phenotypes. Okay, this is the list of the selected some genes and polymorphism responsible for eating practices. Don't worry about these things. You can just see the taste sensations are different, and depends on the genes and where the receptors are, where are the polymorphism, and whether they are normal or they have one copy or two copies of the mutations. Oops. So bitter gene has several variations. So this is just one gene, okay? This is a taste gene, bitter taste gene. And then it has the genotype A, B, and C. And then this bitter food preference is normal. And then this is high, non-testers, because they don't feel it. And then they immediately reject the bitter foods because they have the super tasters. And this genetic bitter taste influence our ingestive behavior, how we eat the food. Like when you go for this TS to eat this receptor, and you can avoid the vegetables, and then you can increase fat and sweet intake, and then you know what happens, how it leads to phenotype. And some of the genotype, this inhibits eating behavior among the women, and they become, then they don't like again certain things, and then maybe they have the eating disorders. And then if they have this genotype, and then it affects their alcohol dependency. So it affects the ingestive behavior, and it affects the taste buds. So this perception of bitter substances are again related with the genes, and they did too many research on that. Okay, so these are the different genotypes, different, and then they have the substances, either prop, this is like PTC, and then quinine or quinine and caffeine, and then when they are liking the taste of the caffeine, they increase the coffee intake and decrease the tea intake. And if they are liking this quinine, and then this is again bitter substances, these are all bitter substances, decrease coffee intake and increase the tea, and decrease coffee intake in the prop one, and then they are more coffee drinker. The role of bitter taste in development of bitter beverage consumption, higher genetically predicted caffeine perception was linked to heavy coffee drinking, defined as more than 4 cups of brewed coffee. Wow, I wish I had one now. Okay, this shows that again tea and coffee contain bitter components that contribute to the pleasant taste. Both drinks containing bitter tasting caffeine, while coffee contains another bitter molecule contains quinine, and the other one, propofol, is just like the tonic water. And here you can see in this research that causal effects of the perceived bitterness of prop, and then if they are the prop drinker, this is the red one, and then they drink more tea, and then if they are the coffee, and then they drink more blue one. See, it's on this side. So sometimes when you have the coffee with the bitter chocolate, coffee makes the bitter chocolate taste sweeter. I don't know whether that makes sense on you people or not. So this is the new research from a pair of Danish scientists. Have found the bitter characteristics of coffee can make a person more sensitive to sweetness. The study found this effect was independent of caffeine and helps explain why many people enjoy the experience of bitter chocolate with coffee, because they think that bitter chocolate becomes sweeter. And coffee and quinine, 2 compounds found in coffee, and they have been previously found in directly inhibit the activity of certain sweet taste receptors. When people were tested after drinking coffee, they become more sensitive to sweetness and less sensitive to bitterness. So the threshold is changing. And then this is the effective in the taste, but in smell, it is not really so much effective because these are more volatile. Okay, so there are certain factors influence the phenotypic characterization of the oral marker of prop. For example, age. By age, your taste buds are decreasing, and then you are not liking the things when you were younger. And gender. When we talk about the woman, they are more sensitive to prop bitterness than men. This is more likely to be a taster or super taster and have more taste buds, and then fungiform papillae. This must be the hormonal changes too. And also they have weight status and obesity. Obesity people become more non-taster. So they don't get the taste and then they keep on eating. And smoking also can influence the characterization of the phenotype. So that we can learn that nicotine is due to stimulant properties. It's an open question. Sometimes the people eat more, sometimes not. Phenotyping and its relationship with this biomarker, nutrition and health are very wide open research areas. So this is the interesting thing. Globally, the number of testers and non-testers varies, have been examined, and then they find out that Japanese, Chinese, and West Africans, non-testers, accounts for as little as 3% of the population. So they are mainly testers. Then you can tell what they are eating. And roughly 70% of the Caucasian people in North America are bitter tasters, and 30% are non-tasters. But when it comes to India, and this is almost a similar culture that Pakistanis have, maybe genetically too, because it used to be just one country. And 40% of Indians were found to be taste blind. And then they can really eat the vegetables because they don't get the taste bitter. And why there's some wide variation between testers and non-testers among people, among the populations is unknown. But maybe this is just a learned thing. Maybe more vegetables are available around in these Southeast Asian countries. Maybe they eat it more, and then it affect the epigenetics, and then this is the effect, but this is good. And there are some research findings about testers and non-testers. And one of the UK study comparing broccoli and white cabbage with the spinach and zucchini. And testers and non-testers agreed the green vegetables were usually more appealing, but testers were more sensitive to the broccoli and cabbage. So they refused to eat these things, testers. And they consumed less vegetables than non-testers, and they did the food frequency according to their culture, and then this was their claim and findings. And tester men more than 66 years old were found with more colonic polyp. And this is again one of the reasons that maybe they were not having enough fiber. And tester men also had higher BMIs and reported less intake of vegetables compared to non-testers. And there's another research. This is non-testers living in a healthy environment consumed more vegetable than testers in a similar environment. So this is the effect of the environment. That's what I was talking in the previous one. And non-testers reported an average of 300 calories a day were taken more because they were eating more. And non-testers preferred reportedly higher intake of spinach, broccoli, and cheddar cheese, whole milk, and high-fat meat, and then also some vitamins. And of course, these are related with the vegetables too. And testers preferred more sweets and sugar. And how it is possible? Because when you are the super tester or tester, when something is sweet, it seems to be sweeter. If something is bitter, it gives you the impression that it is more bitter. So that's why you get this conclusion. And super testers and testers experience more negative sensations such as bitterness and sulfur odors, but expressing the positive sensations of these flavors as sweetness, then, for example, when something is bitter, and especially the medications, for example, if you want to give it to children, if you mix it with the sweet, then they take it more. Maybe that is the reason that they become more familiar with the sweet things, potentially impending on the nutritional balance of one's diet. So some people put the lots of sugar in the coffee or lots of sugar in the tea, then they are able to take it. And nutrients also affects the expression of genetic information, and genetic makeup affects how nutrients are metabolized. Because it's not only like in taken and something, there are other genes, and then this affects your metabolism, whether you can just digest them quickly and you can metabolize them fast, and then the others, they cannot metabolize fast, and then they stay in the system for longer time. Okay, the other one. Okay, this is the gene variance and coffee, and then this is whether the coffee is associated with cardiovascular disease. And we always have some discussion with the cardiologists, right? And some cardiologists say coffee has a protective effect, you can give it. And then the other school says, no, it is not good, then don't give it to patients. So when it comes to genes, again, coffee has this Aqua A2 gene, controls the metabolism of the caffeine. And then it is again gene alleles, and then these are the A, B, C, or you can just tell them that AA fast metabolism, or CA intermediate, or CC slow metabolism. And then this metabolism affects the health of the person too. And coffee does not affect all people equally. So that can be protective, also that can be increase the risk, depending on genetic variation of the caffeine gene, their variant. So if you give them about 2-3 cups of coffee to the fast metabolizer, then 22% less likely to have a heart attack. But when you give 2-3 cups of coffee or tea, of course for tea you have to increase the cups because there's almost half of the caffeine coffee has, and then it increases the risk of heart attack 36%. This is decreasing 22%, and this is increasing 36%. And if you give another 4 cups of coffee, then we can increase the heart attack of the patient, because coffee has effect on the vessels. So if it stays longer in the body, and then if you have these genes that it is slow metabolizing, then you will be in danger. And then you can get caffeine from the coffee, soft drinks, green tea, energy drinks, brewed coffee, and everything. And don't try to have the Turkish coffee because it's really strong. And then fast metabolizers, caffeine consumption may prevent hypertension because it just goes quickly from the body, and stronger physical activities in the sports may have their performance, and then higher risk of low bone density, of course. And then slow metabolizers, and then this is not our concern, and then it has higher risk of hypertension and higher risk of impaired fasting glucose. So this is the diseased LLE. And then this is how the genetic association related with the bitter taste preferences. And then this is the bitter taste, and then these people are the PTC non-tester. So it means they keep less sweet food, and then they take more vegetables, and their vegetable intake is increased because these are the non-testers. They don't find it bitter. And then at the end, they have the less percent of body fat, and then it has a good effect on the plasma glucose. And then this is related with the bitter taste preferences. And then also they have sweet taste preferences, and if the taste sensitivity, sometimes it is mixed with the other things too. And then if you select the sweet food, and sweet food intake will increase, and then it might cause the increased BMI, increased weight, and increase the dental caries. So how these things are affecting the metabotype of the people. And then this is the fat taste preferences that I was talking before too. And then taste sensitivity increases, and then they have the fatty food selection decreases and decrease the fat intake, and then it has a good effect on the heart. And then this is the associated with the ALLE might be the factor disposing to hypertension, and CAD needs to be studied. Okay, this is the metabolite, and then intrinsic and extrinsic factors influence the metabolic phenotype of individuals. Groups of individuals with the similar metabotype, and then the phenotypes are termed metabotypes. Okay, these are the, and we can go either obese people or diabetic or something, and then we can check that they have the similar high glucose level and then high fat level and such, and we can put them in the groups. And then these metabotypes used to deliver personalized nutrition. How you can do personalized nutrition, and then why it is necessary, then it helps you for the early risk identification, metabolites with similar risk of diet-related diseases, and you can see whether the response to dietary challenges and interventions are working okay, and then you can give these similar things for the different metabotypes persons, and then we can see the responses. Maybe you can just reduce the dangers of it, and then you can deliver the targeted advice, and you can target it according to metabotype of the person. And then there is another research, and then this is either you can give the general nutrition to the people. This is metabotyping and group-based nutrition in the context of the contextual population-based guidelines. So for the general nutrition you can give, and most of the public health people are doing it, right? And then, or you can have the group-based nutrition. You can just get the either diabetic group or the cancer group or any, and then you can give them the proper education. Again, see, we all have to give the medical nutrition therapy too. Then that can be either in a bulk or in more specified, and in the personal nutrition, it is adapted to an individual disease risk, lifestyle, and dietary preferences. But it sounds good, but they did further research, and this is from the American Dietetic, and this is the Journal of Academy of Nutrition and Dietetics, like and then they combined all the meta studies related with the gene, and you can see that they did the study between the study design with the design A and design B. On the design A, they got the nutrition council in our care. This was the control group. They just give the nutritional advice to the people. And then they got the genetic testing information, and they give again nutrition counseling care. And then this is with the genetic testing, this is without genetic testing. And then in the other group, and then all these studies, they put them in the group, okay? They didn't do individualized thing. And then genetic testing information, and then in both of them, they did the genetic testing information. But one of them, they give the nutrition counseling or care. And then they combine them. And then the findings were very interesting. And then the systematic review examines results from 8 randomized controlled trials, which were synthesized narratively and with meta-analysis when appropriate. In 13 of the 15 dietary intake outcomes reported, there were no differences in intake between participants who had genetic information incorporated into nutrition counseling or care compared to those who did not have genetic information incorporated. Limited evidence suggests that carriers of high-risk genetic variants were more likely to improve dietary intake of certain compounds than carriers of low-risk genetic variants. So we can just conclude that there is limited evidence for incorporating nutrigenetic testing into standard practice at this time. This field is still in the measuring stage. So I'll just give the take-home message and then I'll wind it up, okay? And multiple genes may contribute to influence sensory perception and dietary intake. Test genes can be modified if you could alter your food preferences to consume healthier foods, and your genes may be more influential in your food choices than you realize. If you dislike the flavor of certain herbs and vegetables, choose others that you enjoy. With so many taste options, you can have to eat foods that if you don't like it, but there are other options, just like food exchange list. If you don't like that one, you can take that one. And if you like asparagus, and you can go for the broccoli or green beans. And taste preferences, this is the phenotypic expression, they can change. It is not really stoned on your DNA. And intervene to a young age, and you can see that I showed you that my grandchild, how the genetics is affecting them, and then don't spoil their genetics. And if child is a tester, teach them to prefer less sugar and salt, and continuously expose them to bitter vegetables. Then they will develop a taste, how the people were developing a taste for the sugar, right? And if child is a non-taster, exposed to healthy fats instead of bad fats. And if the patient is older, then understand their food taste preferences and work around them. And for example, heat destroys some of the compounds of the vegetables or some certain foods, then you can just maybe slightly cook it, and then you can remove the odor or something, and then they can take it easily. And then this is my summary. And Nutritional genomics provides insight into how diet and genotype interactions affect phenotype, and practical applications of nutritional genomics for complex chronic diseases and emergent science. The use of nutrigenetic testing to provide dietary advice is not ready for routine dietetics practice. That's all. And if you have any questions, then maybe I should ask the quizzes first, or somebody will ask quiz.
Thank you so much, Dr. Rezzan. We will have, this quiz first, and then after that, we will have the question answer session.
No, it's not really clear.
Faiza, could you open up the poll? No, I'm asking just Faiza. Faiza
Oh,
Khan.
okay, fine, okay. So I should have closed it.
Each and every participant is being requested to attend the poll to earn credit R and have an e-certificate. It's mandatory.
This Faiza की आवाज
Huh? Now they're asking something?
Yeah, ma'am. They could not hear due to some signal problem.
Faiza, could you share the screen of the poll?
Somebody's answering from panel? Forget it, let them answer.
मैडम आपकी आवाज नहीं आ रही है हाँ जी आपकी आवाज नहीं आ रही है वो डॉक्टर रिज़ान अच्छा तो कितने मिनट्स ओके
What happened?
उसमें she said only 2 minutes wait and then ask question answers.
Thank you so much, Dr. Rezzan. We are having some comments for this presentation. Rabi Anwar is also saying that it is a very interesting and different talk presented by you. It was very informative, and we have got some questions waiting for you to answer. So first we will have a quiz. Participants are being requested to attend the poll. Just wait for a minute. The poll will be launched. And to earn an credit or an e-certificate, you have to attend the poll.
Wait 2 minutes and then very informative and thank you Dr. Rezzan for very extremely informative presentation. Very interesting presentation. Well explained, nice presentation. Very good English.
Questions. Should I answer it?
No.
Oh, this is type answer? We have to type?
No. Answer line.
Oh, okay.
Okay. So there is some technical problem for launching the poll. So we will have the question answer session first. Okay, Dr. Rezzan, are you ready?
Okay.
Okay, the first question is from Rabi Anwar. How will we know which gene is present or dominant in an individual?
Okay.
How can we identify it? Means which gene is dominant?
Okay, genes are not visible. You can tell them from the phenotype. If you make the one black person married with the white person, maybe the child will be black, and then most of the cases, and then this is the dominant gene. Okay?
Okay, she meant whether a person has a risky gene dominant or not, or the normal one. So how can we identify that thing? Means the percentage that I think she meant the FTO one, the variants, the FTO genes risk. So I think
Okay,
she want to ask that one. How will we identify it?
this is you can identify. Huh? Oh, this is you can identify with the genetic test, and then
Okay.
they can do it. Otherwise, you can just give them the, for example, PTC strips, and then they can check it. And then if they are thinking strongly rejecting it, it means they are the super testers. And
Any specific genetic test, Dr. Rizan?
this is, for example, if you are going for the test okay, if you are talking about the test receptors, for example, then you
Okay.
don't need to even go for the genetic test for it, and
Okay.
you can just give them the strip, and then you can tell them to how do you perceive it, then
Okay.
you can get the information. And like as I said, that if these small children, you watch them, what they like, and then there's a very strong genetical effect because the environmental effect is not affecting them yet.
Okay. Okay. Our next question is from Tabinda Ashraf. How COVID-19 virus has changed or altered our genetic material? Is there any nutritional management to help not to get this virus by our future generation? It's COVID related. I will add a point question here for you, Dr. Rizan. In
Okay.
COVID symptoms, there are no taste. The taste is gone. So is it like it is affecting our gene also?
Uh, that can sometimes that can be permanent, sometimes that can be just reversible. And most of the cases it is reversible because it doesn't really affect continuously your histones and epigenetics.
Okay. Okay. Uh, Another question is again from Rabian. Where can we diagnose which gene is down by looking at food preferences and eating behavior?
Yes, she's speaking. Yes.
Can we diagnose which gene is dominant by looking at food preferences and eating behavior?
What?
Then can we diagnose
Which one?
then this is
You're diagnosed with gene is dominant by looking at your food preferences.
And eating behavior, yes.
This is a very crucial thing because the taste is influenced also with the environmental factors. And
Okay.
I told you that when you go for the younger age, then you can get better idea. But later on, depends on the age group and everything. And if you give them the sweets, for example, and then if they are used to getting so much sweets and then their taste buds are blunted, then maybe they will go for more sweets, although maybe they are the super testers for the bitter taste. So you have to check the other informations too.
Okay. Okay. Mehreen Amir is asked what foods help repair DNA?
All the time, yeah.
What foods help repair DNA? Mehreen Amir has asked from you, Dr. Rizan.
then this is what food help prepare DNA.
And I was showing an example between the Mediterranean diet and then the Western diet, and talking about the biocomponents of the foods. And there are so many research, and if you just go for the high antioxidants, and it has a good effect on repairing the DNA. And this is on the epigenetics. But before the birth, we always give some folic acid to the mothers, right? Because this methylation progress, then if you give the folic acid, and then it is going to be affected in a proper way. Otherwise, it's going to cause the problem. So try to give all the nutrients.
Okay. Okay. The next question is from Zarmeen. She's saying salam. And I have a question like all this information is indeed helpful, but what sort of questions we should ask from patients regarding effect of nutrition on genes while taking history, so we can make a proper analysis of their case, as this much information might become overwhelming for them while taking the diet history. Her question is, while taking the diet history, what questions should we ask from the patient to rule out or to dig out any genetic related information?
Actually, you can just get the proper dietary information from them. And you can see what type of thing they are eating, what type of thing they are refusing. And we have to find out these things. Maybe they can eat the proper things, but their parents never introduced them. So this is the loop points in the
Okay.
genetics. That's why they did
Oh,
the research, and then they find out that medical nutrition therapy is more effective than the genetical personalized nutrition at this point, because
okay.
they have to
Okay.
go through the genetic test, and then these are quite expensive too. And of course, the dietitians will be the perfect person to guide them.
Mm-hmm. Okay. Alia Umbreen has a question. Do artificial sweeteners have different side effects on different people? Because some researchers show the artificial sweeteners affect the growth of gut microbiota.
Yeah, maybe they affect the growth of gut microbiota. Maybe that is the bad ones, because it changes the ratio of the good bacteria to the bad bacteria. And then, like the medications, not everybody gets same effect with the Panadol. Some people get affected, and then some people it doesn't have at all. Again, it is the genetical difference. But for the artificial sweeteners, mainly we have to check what type of gut microbiota is increasing.
Okay, that's good. Okay, Zarmeen has asked, I have another question like, which study, source book, author you recommend for the effect of nutrition and genetics? I think nutrigenomics she's asking for. So we can enhance our knowledge. She's asking for some references to have more in-depth knowledge about this topic.
I think I already sent the reading material, and then they can give you further reading. And there are lots of studies going on there.
Yes, yes, I must say.
You
Okay,
just
Mahnur Majid has asked, I want to know how nutrigenomics can affect the metabolic diseases, and what's her opinion regarding the gap that needs to be identified in between vitamin B12 and diabetes with nutrigenomics background in Pakistan?
B12 and
B12 and diabetes.
okay.
With nutrigenomics.
Okay. Okay, again, this depends on the nutrients. Genetics can change the utilization of the nutrients too. And this is different with everybody. Like if you are a fast metabolizer and if you are a slow metabolizer, of course the effects will be different. And this affects for the B12, and then even when you eat the banana, for example, same amount, and it will have a different effect on two people because of the genetic variations. And then there are research again on that. One of my slides was given research studies on that. I think on the FTO. If you just go to that slide, you can get the reference on that.
Mm-hmm.
FTO. Okay.
Okay. Sayeda Mahar is saying, thanking you for sharing a comprehensive view on genetics. She is asking, do we have any labs in Islamabad for nutrigenetic testing?
I don't know. If somebody knows, please let us know.
That will be grateful if anybody has its knowledge. Yeah.
I don't know.
Okay. Murad Baksh is asking why nutritional genomics can't be used in routine dietetics practice as it's immature right now. Means I think he is trying to say the lots of studies are been going on. That is why it's not in practice nowadays. I
No,
think
all
what it means.
findings are there. And if you just read the reference, read the article that I sent from this month, it was released in March, it was released 2021. How we can
Okay.
incorporate the genetics, and
Mm-hmm.
it will give you the all detailed things because it combines all the research together and then they analyze them for this meta
Okay.
studies.
Okay, that's good. So I
Some
would
of
appreciate if each and every part attendee should read out each and every reading material provided by Dr. Rezzan. Okay, Aniba Batul has asked, can you please explain some foods or dietary habits that can improve our genetic makeup? It's a
okay,
repeated question.
can go for the healthy diet and then it goes past your next generation. Increase your exercise, it can pass the other generation. And then, because whatever you are doing, it's really affecting the babies just before the birth in the uterus.
Okay,
So, and just healthy habits,
healthy
lifestyle,
habits and lifestyle. Yeah, healthy
yeah,
lifestyle. Yeah.
sleeping and,
Mhm.
all those things. I don't want to go through one by one.
Yeah, sure. Okay, Aruj Ijaz has asked, from what fetal age does the taste receptors develop? Can receptors be affected or modified during early childhood? Even genetic makeup is different.
Yes, it can change. It can
I
change.
think taste can be developed and changed at any age,
Just like
as far as I think.
and you can see that some of the ladies, when they are feeding their children, they say that when I eat garlic, the child likes it more. Always think that that is not true, but you can hear it from the people.
Yeah, I can see.
But
Okay. Rabian is asking, how can we personalize a nutrition plan looking at the phenotype? How we will diagnose if a client is super taster, or non-taster.
looking at the phenotype, you'll be seeing that people will be having either slim or so much fat, and we can tell that one. And when you go through the dietary habits of the patient, you will get an idea whether the person is super taster, or non-taster. And especially some of the patients, when you tell them about the vegetables, you can see their face. But in the younger age, it is more stronger. And you saw that how my grandchild was throwing out the
Mm-hmm.
chocolate
Yeah.
and eating bitter coffee and lemon. So,
That was the lovely part of your presentation.
so it is more dominant, and later on, the effect of the environment is there, so it affects the food choices too. That was all about this presentation.
Okay. Dr. Rizan, with the thing you said that super taster eats less as compared to the non-taster, they eat more. But what I feel
Super
like, if the
taster,
food
no, no, no,
taste,
super taster refuse the bitter food, and then they
okay,
go more sugary and fatty food.
okay, got it. Means the super taster are at more risk, I must say.
Yeah, they refuse to eat the vegetables, bitter ones, and such things. Because
Okay.
actually, most of the babies are having super tasters. Just the nature wants to protect them. Like the sour things, they throw immediately. But my kid was the different one.
Okay, we have another question. It was from an unknown attendee. During MNT management, how we can find out genetic behavior of patient? It's again from the dietary recall. I must say, if Dr. Rezzan, could you please?
Yes, yeah. Okay. How we can find out genetic behavior of the patient by asking questions. Yeah, by asking questions, and then you have to just evaluate it, and you have to work like a radar. You have to screen everything what they are saying,
Yeah, yeah,
and
I
you can
must.
find out
Okay,
whether that's
say the
or this
yeah.
because of the environment.
Okay, Sayyida Aimer Rizvi is asking, can we suppress disease-based genes through genetically modified foods, GMO foods?
Genetically modified foods. Okay. Yeah, genetical disease. For example, if the patient is having phenylketonuria, right? There is no enzyme developed there. Then you can just change the diet, and then you can give low protein, and then you can minimize the phenylalanine, and then you can add a little bit of tyrosine and cyclic, and then you can do that, but not with the genetically modified foods. Then you can give the supplements, and then I don't know what is this genetically modified foods, but for the nutrition plan I'm talking about, then you can give mainly for the, if you are reducing the protein, then you are going to give the more fruits, vegetables, and then you can see to solve the problem. And if there is folic acid deficiency, then you can just add it, and then it can continue even after the birth if we can prevent this bifida and such things. I think that's what it means.
I think so. The GMO foods meant
Yeah,
the same. Yeah.
yeah.
Okay, Murad Bash is asking, set point theory says that obesity is in your genes. Without affecting your genome, it could not be settled. What do you say?
What do you say, Murat? What do you say?
He has asked you
We
that
can
yeah.
change these things by changing the lifestyle, right? That was in one of my presentations again. Just see that scale, how the polymorphism is affecting the genes and then the environmental effects. So you can be either going on the right side or the left side of the scale.
Okay. Okay, Dr. Rizan, with more interest in this topic, Faiza, our president, PNDS president, has asked that we can plan a comprehensive course on this topic with you as a means chair, and we were for our members, as members are also showing interest in this topic, and I must say, इन द मीन वाइल मैं अभी इस चांस को अवेल करूं और अभी आपसे कमिट करवा लूं so we will be having a comprehensive course with you on this topic.
Okay.
You will be I think you will be ready for it. We'll be pleased to have you.
Interesting. That's it.
So, okay, that's all from the question answer session. Okay, Faiza, you can start the poll.
मिस फायदा की आवाज नहीं आ रही.
I even can't hear her. Faiza, we can't hear you.
मैम some error.
Mr. Salman will launch the poll. within few seconds, and all the attendees are requested to attend the poll. It's mandatory. Okay, wait. Sarosh want to ask something. Sarosh, can you type? We are already done with our question answer session. Dr. Rizan, for the meanwhile, the poll is being launched. I have received another question. I'm sorry if you could answer it. Alina Omi has asked if someone have might genes of some sort of allergy, but after teenage gained that allergy. Is it possible to suppress that specific allergy by food management? It's related to any.
Maybe food management, but we can see what you are allergic and check your stress level too. Sometimes when you are under stress, you are more prone to get specific allergies too. And the best thing that you can keep a diary and then see what you are allergic. And then we can use the elimination method.
Okay, got it. Thank you, Dr. Rizan. Now the poll is being launched. All the attendees are requested to attend the poll.
They are asking, these are all different questions.
Yeah. Oh, when you die.
Sitting makes me tired.
Is the poll done? It's still in progress.
Rezzan asks for share information about nutrigenetic testing. Where is the Tramba nutrigenetic testing available? I don't know. Not please arrange a workshop on.
Yes, I did it, right?
Yeah.
I should. You should have just seen it. I put it there. Consultation and labor room.
90 participants. 90 candidates.
That was a large number, Dr. Rizan, I must say.
Yeah.
Mashallah.
90 participants.
And it's our day, everybody must have been so busy, 11 o'clock.
So looking at the interest of our, our members, that is why Faiza has asked to ask you to have to plan a comprehensive course in future on this topic. it's a,
Maybe we can have it in the conference.
Does people affect all people equally?
It was difficult to digest that.
Let Mapara join this. Let Mapara join this. She said,
Mahpara is asking, does all people affect all people? Does it affect all people? I think she's asking for the nutrigenomics.
Oh yes, it does. Because everybody has some variance. Whether the dominant one or the recessive one or the mixture of it. Question? I didn't turn that.
no, this is
Okay. So it's over.
Poll is still in progress. The participants are taking time. Pretend the poll. That's good. Okay, see, one of our participants has asked to plan a workshop on this topic. It's on workshop.
Good idea.
Standard individualized questions.
Yeah, this is the last question.
Yeah.
He's asking.
I mean, how many questions you send? How many questions you send?
8. It's the 8th one.
Yeah. Class
Okay, they are on 6-1. The attendees are on 6. One of our attendees has offered that I'm all available for the assistance to the workshop on nutrigenetics.
And Navish said,
Okay, we are all done with the poll. So thank you so much, Dr. Rezzan, for having such a very in-depth knowledge and informative presentation. It's really a new thing, not very new, but new for some of our new dietitians, the junior ones, and still I must say, the researches are ongoing and all. It's really an interesting topic, and we should put it in our practice, routine practice, as one of our attendees have also asked question about it. So I would request all the participants to read out the reading material provided by Dr. Rezzan. The PPT also will also be shared in the PDF version. You can find it on our website. It will be uploaded soon. The e-certificate will be circulated within the 15 working days. You will receive on your email. And thank you once again, Dr. Rezzan, on behalf of PNDS. Wish you all happy, healthy Ramzan and safe Ramzan.
Thank you. Thank you. Thanks for joining it. Finished?
okay, Miss Sabine.
Hello.
Oh, I thought
Okay, Miss
everybody
Sabine.
was gone. Okay, thanks a lot.
Miss Sabine? Yes.
Gone, I think.
Gone it.